Thank you, Marina. And our last talk will be from Rebeca de la Fuente. So, the floor is yours.
Well, in this work, we wanted to answer two different questions from a comparative perspective. One of the questions that we wanted to answer is how the genome architecture is in multicellular organisms and how it differs from unicellular organisms. And also we wanted to answer the question of how is the genome architecture related to the alternative splicing, which is the mechanism by which different protein isoforms are generated from a single gene. Well, okay, we can wait. Okay, now we have it. So, in order to answer these questions, we identified a mathematical model that captures the relation between the gene content and the percentage of coding DNA present in genes. And we found that there is a transition at the origin of multicellularity where prokaryotes and unicellular eukaryotes, whose genomes are mostly composed by coding DNA, show a linear scaling as expected. But in the regime of multicellularity, we observe a sublinear scaling, which means that the expansion of coding DNA is slowing down as the gene content increases. We also compared the genome size to the gene content and observed that plants is the taxonomic group that deviates from the general trend. Well, we also wanted to understand if alternative splicing has something to do with these relationships and if it is exclusive to multicellular organisms. So we developed and proposed a novel genome-wide measure of alternative splicing per species. Then we obtained a single value per species, which allow us to perform a comparative analysis, taking into account more than 1,000 of species. So we compared the alternative splicing for different taxonomic groups and also compared to the genomic variables. And in general terms, we found that the alternative splicing is in general maximized in genomes that have minimal percentage of coding DNA and in genomes that have intergenic percentage at about the 50%. Also, interestingly, we found that there is a high correlation of alternative splicing with the genomic variables only in mammals and birds. So that's all, and I will be happy if you would like to discuss these results. So come to see me at the poster number 12. Thank you.